Poseidon is a phase 1 randomized phase 2 trial of tamoxifen and taselisib or placebo in patients with HR-positive, HER2-negative metastatic breast cancer. So as we know, the inhibition of the PI3K pathway may refer to endocrine resistance, and this concept has been proved in several trials like Sandpiper and the Solo-1 trial that led to the approval of fulvestrant and alpelisib. So this trial is testing the combination of another endocrine agent, tamoxifen, with a drug that is a PI3K beta-sparing inhibitor, taselisib. So Poseidon is a phase 1b randomized phase 2 trial. The phase 1b part tested the combination of taselisib and tamoxifen in patients with ER-positive, HER2-negative metastatic breast cancer. The results were published a couple of years ago in Clinical Cancer Research. And the trial showed that the combination was tolerable and had some preliminary signs of activity. Now we are presenting the randomized phase 2 part of the trial. So in this portion of Poseidon, patients with endocrine ER-positive, HER2-negative metastatic breast cancer that were resistant to prior endocrine therapy and had up to 5 prior lines of chemotherapy for metastatic disease were randomized 1-to-1 to receive tamoxifen and taselisib at a dose of 4 milligrams a day, or tamoxifen and placebo. Patients were treated until progressive disease, and upon progressive disease, patients on the control arm could cross over to the treatment arm. The primary endpoint was progression-free survival, and key secondary endpoints included overall response rate, clinical benefit rate, safety, and overall survival. The trial was powered to detect a hazard ratio of 0.64 between the arms, with a power of 90% and an alpha of 0.2. Unfortunately, with the pandemics, we had to interrupt the enrollment of the trial, decreasing the power of the trial to 83% to detect the same hazard ratio of 0.64. So 152 patients were included in Poseidon. Mainly, these were post-menopausal patients and who had received a median of 2 prior lines of chemotherapy for metastatic disease. We could test the PIK3CA mutation status in about half of the population until now, and we found a proportion of PIK3CA mutation around 25%. Poseidon met its primary endpoint of increasing PFS in this population of patients. The hazard ratio for PFS was 0.63, with a statistically significant p-value of 0.018. The median PFS in the taselisib plus tamoxifen arm was 4.8 months, and in the placebo plus tamoxifen arm, it was 3.2 months. And the benefit was observed in all subgroups independently of the presence of a PIK3CA mutation. Patients less pretreated seem to derive more benefit from tamoxifen. Regarding the key secondary endpoints, overall response rate was numerically higher with taselisib, although the proportion of patients with partial response was small, 9.2%. And clinical benefit rate was also numerically higher in the taselisib arm. There were no differences in overall survival, but of course the results, the data are still immature, and I remind you that the trial had a crossover design that possibly hampered the overall survival results. Regarding safety, the toxicity profile of the combination was very consistent to what is described in the literature regarding taselisib. Most common adverse events were hyperglycemia, diarrhea, nausea, mucositis, and elevated AST and ALT. And of note, patients receiving taselisib had a higher proportion of adverse events overall, and also grade 3 and 4 adverse events. 43% of patients receiving taselisib experienced grade 3 or 4 adverse events compared to 5% in the placebo arm. Yeah, so Poseidon met its primary endpoint. So PFS was higher, statistically significant higher in the arm of taselisib plus tamoxifen, respect to placebo plus tamoxifen. However, the tolerability of the regimen is poor. So I think the results of Poseidon reinforce the concept that combining endocrine treatment with agents that block the PI3K AKT pathway is a valid strategy. However, we need new biomarkers that can identify patients that can derive most. The combination will not be further explored, but I think that the concept is valid and should be further studied. Yes, so currently we are undergoing an extensive biomarker analysis, not only the PI3K mutational status, but also incorporating ctDNA analysis and other biomarker analysis to figure out if there are particular patients that can benefit from this combination. And again, this combination in particular will not go further because, as you all know, taselisib, the development was discontinued. But I believe that the concept of PI3K AKT inhibition plus endocrine therapy is a valid strategy in ER-positive, HER2-negative metastatic breast cancer. And we are definitely working in continue pursuing a trial in this area for the next future. So Poseidon is a collaboration of three main institutions: the MKI Cancer Institute in the Netherlands, the Cambridge Cancer Center in the UK, and the Vall d'Hebron Institute of Oncology in Barcelona. And this is the result of the collaboration of a huge team and the Radar Consortium, the Genentech, and a lot of people that were instrumental to make this trial a reality, and who I would like to thank.